Thank you. Yeah, so thank you for the invite. As Optical said, I'm Dr. Wright, and I'm based in the Optics and Photonics Group in the Department of Electrical Electronic Engineering and at the University of Nottingham. So just a little bit of an introduction to me. So the Optics and Photonics Group is quite a large research group in the engineering faculty, and about half of us are based over in the life science building. So what that does, and it's great, is it gives us access to lots of cell culture facilities, great access to collaborators and people that we want to work with in life sciences and in medicine. And that's where a lot of my research has been over the last 20 years, has been developing some novel optical systems and instruments that can help life scientists and clinicians going about their work and imaging cells and tissue. My kind of small team within the group is probably about 2 postdocs, 3 PhD students and a cell culture technician. That sort of fluctuates from time to time. And over the years, I've worked on some different techniques such as adaptive optics, aberration correction, confocal microscopy, light sheet microscopy, and quite a lot of optical trapping as well. This here, hopefully I can get it so that you can see great, yeah, so hopefully you can see my pen now. This is one of the latest instruments we've built, and this is an optical trapping system combined with a light sheet microscope. And today I'm going to talk a lot about some of the work that I've done in adaptive optics. So I'm going to move away from optical trapping and talk about some work I've been doing on adaptive optics on and off over the years. So the kind of premise for this webinar and the sort of challenge Optical sent me was to think about where I think this field is going in the future. And I think when you develop optical instruments, there seems to be this underlying push to always try to image your samples deeper, to image them faster, to look at smaller sample features within your sample, and try to do all of this with minimal sample depth. So that's the kind of big question, and where we would like to all get, and a lot of what the technology development is pushing towards, either one or two or a combination of these kind of things. So today I'm really going to concentrate on some recent techniques over the sort of last 10 or so years that allow us to image smaller things, and I'm going to spend the majority of my time really on sort of up and coming techniques and things that are out in the literature at the moment about how we might try to image deeper into our sample. That's where I have a particular. There's some recent work that's taken place over the last 10 years about how we push the resolution limit. Some of this will probably be quite familiar to you all, but I think it's quite a nice example of how optical microscopy can help, and how we can really, there's always new and interesting things to do, and how we can really push what we're able to do with our instruments as we design them. You'll be familiar with this guy, hopefully. So this is Ernst Abbe. So he set this diffraction limit, this equation that a lot of us are very familiar with, which tells us how small what's our smallest object that we can resolve within our microscope. So if we have 2 objects in our imaging system, how close together can they be that we still think of them as 2 objects, and rather than them looking as 1 object. So d is this distance, lambda is the wavelength of the light that we're imaging with, and this part at the bottom here is the numerical aperture of our microscope objective, or the refractive index of our medium, and the angle here created by our objective. And for many years, it was thought that this put the limit on. It was thought that actually we couldn't, with optics, we weren't able to image anything that was closer together than this distance. And what does this really give us in terms of biology? What does this let us image? So depending on the lens you use, the numerical aperture that you're using, and the wavelength that you're using, Abbe's diffraction limit tends to sit around a few hundred nanometers. So that means that we can image hair, we can image cells, we can image bacteria, and actually we can image components of cells and components of these objects. But what we can't do is we can't get to proteins, we can't get to single molecules. And it was felt that these were kind of beyond what was achievable with optical microscopes. And then in 2014, these 2 people here won the Nobel Prize for Chemistry, and that was for developing new types of optical instruments that were able to beat this so-called diffraction limit. And they formed this field of super resolution microscopy that a lot of us are quite sort of used to that term now today. I always think it's quite interesting to note that they won it in chemistry. In many ways, a lot of these techniques that were very groundbreaking at the time came from developments in the fluorophores and the fluorescence probes used to do this imaging, and I think that's reflected in the fact that they won the prize for chemistry. So how do some of these techniques work? So STED is one of the techniques. There's 2 that were actually that won this prize. So STED is stimulated emission depletion microscopy, and this is a way of trying to image beyond the diffraction limit. So essentially, you start by illuminating your sample with a diffraction limited spot. So that's shown in figure 1, and this produces a population of excited fluorophores, and their distribution matches the distribution of your excitation beam. Your sample is then excited with a second beam that has a donut-shaped profile to it, and this is called your depletion beam, and this quenches the fluorophores, which it illuminates and sends them back to the ground state. So essentially you quench all your fluorophores in this ring, and you're left with just collecting fluorescence from this central spot, and therefore you can see if this is your diffraction limited spot, you'll end up able to correct fluorescence just from a region that's a small proportion of that diffraction spot, by essentially switching these fluorophores off and controlling the fluorophores in your system. So SED works by scanning these two beams across your sample, and in doing that you're then able to build up a two-dimensional image, and you step in Z and you build up your three-dimensional image. This is now a commercial instrument. You can definitely buy one from Leica and possibly from other manufacturers out there, and you can just see some nice comparison images of some confocal microscope images along with some SED images, and you can see the advances that were made. And this is a paper published by the Stefan Hell group who developed this method, and you can see here really pushing this limit. You can see that they're imaging objects that are now 20 nanometers and much smaller than the diffraction. The other technique that was used to try to beat this limit was a sort of form of localization microscopy. There's two different acronyms used here, and I think that's just to do with the properties of the fluorophores that are used in the different cases. You sometimes hear it called PALM, and you sometimes hear it called STORM. And the basic principle behind these techniques is the fact that if you image a single molecule, you can fit the center of that molecule, although you might not be able to resolve that molecule. So you can see here this kind of spot. So this is fluorescence coming from a single molecule. You can't resolve it, you can't see the edge of where that molecule is, but you can do a fitting procedure to try to localize the center of that molecule. And that's what these localization techniques involve. So essentially you excite your sample with a weak pulse of light that activates just a fraction of your fluorescent proteins. And the idea is that the distance between any of your two proteins that are activated is greater than the diffraction limit. And then you can then work out the center of these fluorophores, of these proteins that you have activated, and you can map them to your image. And you take many thousands of images, and you've got slowly build up this picture of where the fluorescent centers are located within your sample. And here's some images here that are taken using a palm microscope and the membrane of a lysosome. And you can see the conventional fluorescence microscopy, the localization microscopy, and then the zoomed in section here. And if you look at this final image, it's quite nice. That scale bar is showing you what the normal diffraction limit would be for that instrument. And you can see features far smaller than that in there. And again, some images taken showing a sort of T-storm version of this, where you can see some incredibly small features in your sample. So that's really where the field's been pushed in the last sort of 10 years or so in terms of the smallest features that we can image. Now I want to talk a little bit more about how we can extend the depth at which we can image within our samples. So let's just talk a little bit about the problem, really, if you like, if you try to image at depth. So the problem really comes from absorption of your light within the material and also scattering of your light within the material. In biological samples, we tend to be much more concerned with the scattering coefficient. That tends to be the dominant effect over absorption, but absorption is still present. And we can characterize this by thinking about the mean free path of our photons. So that's the average distance a photon will travel between two consecutive scattering events, and then also the transport mean free path of our photons. So the transport mean free path is quite an important one when it comes to imaging, and that's the mean propagation distance that a photon takes on average to lose any relationship to the propagation direction that it had when it entered the tissue. So you can see that in this nice image here. So this come from a review paper that's quite a useful read if you're interested in this, and you can see this mean free path here, and you can see your transport mean free path. And by the time you get to this point in your image, essentially the direction of these photons have no relationship to the direction that they had when they first entered the tissue. And we also talk about this reduced scattering coefficient, and this takes into account this property G. And G is basically a property of your material, and it tells you how much of the light is forward scattered in that material. So it scales this relationship depending on how forward scattering is the material that you're looking at. And this is different for bone, for example, compared to blood or tissue. This is very much sample dependent and tissue dependent. And it's called the anisotropy function, and it's the degree of forward scattering. And if we assume that this dominates over absorption, we get this relationship here for our transport mean free path. It's important to note that this scale along here, because it depends on G, it depends on the tissue that you're imaging, and it also depends on the wavelength of the light that you're using. So just bear that in mind. Another way in which we can look at this scattering is we can consider what's happening to the light as it travels through our tissue. So we've got our input wave vector here with the signal strength eta, and k our wave vector, which is telling us what our direction is. If we have a target object sort of buried within some kind of scattering material, what we're really interested in, what we would like to detect, is this red photon coming out, and that's a single scattered wave that's just been scattered by our target of interest. But actually, in the same direction as that wave, there's many other scenarios that can occur, and many other waves that can be present. There's this TM wave and BM wave, which essentially have taken the same amount of time to pass through the tissue and have also come out at the same direction. And then there's this green wave, which has taken longer to come out the tissue, or possibly shorter to come out the tissue. This has a different time of flight to these three. But essentially, all of these waves here have undergone multiple scattering events, and therefore they create a kind of background and a sort of blurring of our image that means that we can't just detect this single scattering event, which is the wave essentially that we're most interested in trying to detect, and the wave that's got the information about the sample that we're trying to retrieve. Another way of thinking about that, again, this is taken from a review paper that I would highly recommend, is to try to think about the relative intensities of these different types of scattering events as you go deeper into your sample. So as I said, we're really mostly concerned, we'd love to be able to retrieve this information from the single scattering event that's showed here in this sort of reddy pinky colour. But as we go deeper into our sample, the signal that we detect becomes more and more dominated by these multiple scattering events and events that have occurred at different times of flight and from different parts within our sample. And that creates a background, gets in the way of us finding the signal we're interested in. I just wanted to show you this image as well, because I think this is a really good way of just sort of highlighting the problem. So when I first started working on adaptive optics, I was at Strathclyde University working with John Gherkin, and these are some images that he had taken of some Philadelphia cream cheese with a 2-photon fluorescence microscope. And you can see here this nice image that you can get at 10 microns, and you go 90 microns into your sample and really no longer is anything resolvable. So this is the problem really, and this is what happens as you try to image deeper. Your image blurs and you no longer really know where your photons have come from within your sample. The way this is often tackled is a sort of a series of different techniques that are all called wavefront shaping techniques essentially. And the idea is that you have an optic in your system. It might be something like a spatial light modulator or a deformable membrane mirror, and you use that to shape the wavefront of the light to change the local phase across your wavefront of the light as it enters your tissue. And if you can predict that correctly, shape that correctly, what you can do is in your sample, you can create a focus at the point of interest that you're interested in imaging. So the idea is to try to shape the wavefront in such a way that you create a focus within your sample at a site that you have determined. The big challenge here really is to know what shape you want to put on your SLM or your deformable membrane mirror, and how you want to control this and how you're going to determine the shape required. So that's really the challenge with a lot of these techniques. And as I said, the two different optics that are on devices that tend to be used to shape your wavefront, you tend to get people using either deformable membrane mirrors or spatial light modulators. So deformable membrane mirrors is a very thin reflective membrane. It often works electrostatically, where you have a layer of electrodes and you can apply local voltages to those electrodes and shape your membrane accordingly, and therefore you can add delay to your beam at different places along the way, depending on how you shape that. A spatial light modulator has a lot more pixels, so a lot more locations where you can shape your beam. And again, you have an array of electrodes and you have this liquid crystal layer. And by changing your voltage, you can change your orientation of your liquid crystal and hence your local refractive index and the local phase of the light as it hits that pixel. Generally, the comparison between the two is deformable membrane mirrors tend to be a lot faster in their update rate, so that can work if you're using an optimization algorithm type approach. SLMs are slower, but SLMs often have a much better resolution. You're often talking about 256 by 256 pixels or even more than that. So therefore, you can get lots of local changes in your wavefront as you expect. The first kind of way people really approach this problem was to borrow some techniques that are often used in astronomy and techniques called adaptive optics. So here's just some images here showing how this is often used in astronomy. And here they're trying to correct for the sort of layer in the upper atmosphere where you get lots of temporal variations in refractive index, and that distorts the beam. So what you would typically have in an adaptive optics system is you would have a wavefront sensor, a control system, and a wavefront modulator. So you can see here you have your aberrated wavefront coming in. So here this would be coming back from the star, for example. It would go off your deformable membrane mirror or spatial light modulator. You would use a wavefront sensor, maybe a Shack-Hartman sensor, to measure that wavefront. And then you would have a control loop feeding that back to your modulator, and eventually you would correct this so that you ended up with a nice plane wave coming to your lens that was focused on the camera, and that would allow you to get a nice image of your star. The other way you can do it is you can read a fitness value back, and your control loop can rather than actually measuring and sensing the wavefront, you can read a fitness value back that you believe leads to a better quality image. So often people would use intensity here, so they might read intensity of a point in their imaging, and they would rapidly change their wavefront to try to maximize on intensity, and in that way they would correct for their aberrations. And this kind of approach of using a fitness value tends to be more suitable to biological imaging, because often you don't have a known point of reference. Going to use a wavefront sensor approach, you need to have something in your sample that is hopefully a point source, or if not a feature that you know what it should look like, so that you can correct this accordingly. I'm going to give an example now of an experiment we did a while ago where we used adaptive optics in a CARS microscope, just to show you how this can work and how it can be effective. So for those of you who don't know, CARS stands for Coherent Anti-Stokes Raman Scattering Microscopy, and essentially you collect the Raman scattered photons from your sample, which gives you a kind of chemical signature of the molecular vibrations that are taking place, so you can identify different components within your sample, and it's a non-linear process, so the process only occurs at the focus of your laser beam, something like multiphoton microscopy. You have two beams in CARS microscopy. You have a Stokes beam and a pump beam, and you excite your molecules, you excite these vibrations in your molecules at the beat frequency between these pump and Stokes beams. And what this does over normal Raman microscopy is it gives you an enhanced signal, and it reduces your background. You force these molecules to go at a particular resonance. Some of the advantages of using CARS is that it's a label-free, chemically specific imaging, so you're not putting any fluorophores into your sample, and because of that people think of it as minimally invasive. You tend to use longer wavelengths, and I'll talk a little bit more about that in a minute, but that has some benefits when you want to image deep. It's non-linear excitation, therefore it's localized, and it gives you a nice three-dimensional image, and the near-infrared light tends to help you go deeper into your sample. This is the experiment that we did. We had our deformable membrane mirror. We collaborated with a group in Harvard who'd built this nice CARS microscope, and we took our deformable membrane mirror and our optimization software, and we put that in their system to see if we could improve on their images. So you can see here that you've got the pump and Stokes beam coming out of the OPO. We've got some relay lenses, a polarizing beam splitter cube, and a quarter wave plate to get the light to the deformable membrane mirror, a scan head, and then the light goes in the back of the microscope. And then the control loop reads the signal on the photomultiplier tube that collects the CARS signal, and it reads it back to some A to D cards, and it can then optimize on the intensity of that signal. And that's how we go about trying to work out what shape we require on our deformable mirror. The algorithm that we used at that time was a fairly sort of straightforward random search algorithm. So essentially we had a deformable membrane mirror that had 37 actuators. Our algorithm would choose an actuator at random. It would change that value on that actuator by a random amount. It would look at the signal coming back from the photomultiplier tube, decide if it had increased or decreased. If it had increased, it would accept the change. If it had decreased, it would reject the change. And then it would go back around that loop again. And if it had tried this loop 50 times and hadn't managed to make an improvement in signal intensity, then it would say that it had reached the solution, and it would finish the optimization algorithm for us there. So what we did is we split our aberrations up that we were looking at. We looked at our system aberrations. So these are aberrations coming from the optical microscope. We looked at depth-specific aberrations and recorded some lookup tables there. And we looked at more location-specific aberrations, thinking about how our aberrations might change across our field of view at particular depth in our sample. So the system aberrations, for this we just put a cover slip on our microscope, and we were able to look at the non-resonant CARS signal from the air-glass interface. And you can see our uncorrected image here on the left and our corrected image in the right, and this sort of equated to about a 2.2 times improvement in intensity. You always get this non-uniform intensity distribution in cars, and that's because you've got two different wavelengths overlapping and you get a chromatic walk off at the side. But you can see that the deformable membrane mirror correcting for our wavefront was able to improve our signal here. Then we made up a sample of some agarose gel with 10 micron diameter polystyrene beads embedded within it, and we found beads at different depths in our sample. We focused on them, and then we optimized the intensity of the bead at that point. And you can see what happened here. So in doing this, we were able to build up a set of lookup tables, so where we would optimize on the bead at a certain depth, save the shape that we'd found on our deformable membrane mirror, and then we could apply that shape to different depths within our sample. So this was without any correction. So you can see here that we get a reasonable amount of signal at the surface, and that falls away as we go up to sort of 700 microns deep into our sample. And then these are with our different lookup tables. And you can see as you go deeper, these lookup tables that were determined at deeper within the sample kick in and start improving our signal. So this is just an image here taken of a bead just under 600 microns deep into our sample, and we're applying a lookup table recorded just lower than that, and we were able to get a maximum signal enhancement of about 10 times. We then moved on to a sample of chicken muscle, white chicken muscle, and looked at signals coming from the lipid droplets within that. And again, we were able to park on one of these lipid pockets within that and optimize our deformable membrane mirror and improve our signal. And this is looking at 260 microns deep into a sample there. And using this image, we could work out how much our enhancement changed across our field of view. So the way we do these measurements is we park our laser beam, or we scan a very small region of the interest, and we read in the intensity from that region. So we were interested to know how much intensity changed as we moved across our field of view. And you can see that drops off with time, and we no longer make, as we get further and further away from the point of optimization, the correction we make just for our system starts to get comparable to the correction we make for our system and our sample. And there's another technique out there that was developed by the Oxford group, kind of a similar sort of time to when we were doing this work. And it's a different approach. It's a nice approach to correct for aberrations. And they use these Zernica modes. So this is what I'm showing here in this triangle. So Zernica modes are a really accurate way of representing kind of common aberrations that are found in microscope. So you can see spherical aberration here, tip tilt, defocus, and different aberrations that you find present. And what they do to correct, they read in intensity again, but they apply different shapes to their system. So they have an initial aberration, so they record the intensity at initial aberration. They take each Zernica in turn and apply a positive amount of that Zernica, a negative amount of that Zernica, and then they have 3 points with on this parabola with which to fit, and then they can find the center of that. So this has some benefits in that it's a lot quicker. You don't have to go through all the different changes on the mirror, but it does rely on a level of aberrations that you get a nice parabola in your search space. So there's some sort of pros and cons to the different method, and this is a nice method that was put together by the people in Oxford. And we had a little go at this a while ago, so you can see an aberrated focal spot and then a corrected aberrated focal spot by correcting each of these Zernicas in turn rather than correcting for each of our actuators in turn. And one thing that we've been wanting to do for a while was to think about how we can try to speed up some of these corrections by putting some a priori knowledge into the system. So a PhD student who just finished last year developed some ray tracing simulations to try to simulate our sample and predict the aberrations present. And from that initial prediction, initial best guess, if you like, run an optimization from there to see if we could speed up the process. And we were able to significantly speed up that process. So I've talked a little bit about how we can try to go deeper by correcting for what we tend to term aberrations, but I wanted to move on to really thinking about scattering. So it's a bit of a grey area in many ways as to where we talk about aberrations and when we talk about scattering, but aberrations tend to be referred to as lower order perturbations. So this is where you've got a kind of a bulk change in refractive index within your sample and your wavefront propagating through your beam, and you kind of delay a sort of section of that wavefront by a fixed amount, whereas a scattering is much more higher order, and individual photons are changing direction due to the size of the features within the sample, and by the time you get to this transport mean free path, the direction of the photons are completely random with respect to each other. So there's different things are needed in these different cases, and aberration correction really will only ever get you so deep into your sample. And at some point, you're going to need to start thinking about these kind of scattering events as well. So adaptive optics, the sort of examples I've just described, work well for aberrations. I now want to talk a little bit about some of the papers I found recently in the literature that attempt to correct for scattering and to find techniques to overcome some of the scattering problems. So here's a few examples. So there's some nice approaches out there that they talk about focusing multiple scattering waves. So if you take a nice simple imaging system, a lens system, you have your object plane and your image plane, and you're able to image your system on your camera. If you then put some scattering medium in the way, some highly scattering medium, a thin slab in the way, what happens is by the time you get to your camera, if you've got a coherent light source here, you end up with a speckle pattern on your camera that you're no longer able to recognize your object. So the idea of this focusing of multiple scattering waves is that you correct for the local phase incident on the wavefront, so you make lots of small adjustments to the phase across your spatial light modulator. And what you're aiming to do is you're trying to change the direction of the wave locally so that when it arrives at the camera, you get constructive interference, and therefore you can bring your get a focus on your camera because you get all these individual speckle points coming together to have constructive interference on your camera. And the intensity of one of these speckles by this kind of technique can be enhanced up to about 100 times. So that's basically making all these speckle points interfere at a point within your camera when the appropriate phase pattern is applied. And the result is a bright spot on the camera with a contrast proportional to the number of controlled elements on your spatial light modulator. So this is really where you need to switch, for example, to using a spatial light modulator as opposed to a deformable membrane mirror, because you want to make lots of local changes depending on this scattering properties of this material. And the SLM essentially acts to transform the scattering medium into a scattering lens, and what you do is you end up with a kind of 2-lens imaging system. So some focusing is happening with your SLM as well. This is different in principle, although often an optimization algorithm is run to help you decide what pattern you want to put on your spatial light modulator. This is different in principle from adaptive optics because the idea here is to constructively interfere many different paths, whereas adaptive optics essentially aims to correct all the optical paths so that they have the same length. So there's a slightly sort of different ethos behind how you're doing this correction. What this method does do is it assumes that the detector can be placed in the plane behind the sample. So there's something and sort of some translation that needs to be worked on here for this kind of technique to be applicable maybe to biological imaging, but it's a nice approach. So here's some results from that paper. So this is a sort of standard speckle pattern. This is when you're imaging through a highly diffuse scatterer, and this is looking at a point source. So if you correct locally for the different phases on your spatial light modulator, this is the phase map on the spatial light modulator after correction. So you can see that all the pixels were required, that resolution was required. You can bring your pattern together to get constructive interference and therefore a nice imaging point spread function. This is another application here where there's a transmission plate with the letter A in it. So you can see here with just a conventional image going through this diffuser, this highly scattering medium, you're not able to see the A at all. You can't see anything in your image. If you optimize your spatial light modulator, you can see the letter A quite clearly. There's a bandpass filter in place, and that's to do with the sort of different chromatic aberrations that come from correcting for phase on your SLM. And if you remove that, you can see that your image blurs. And in this case, it was really nice because this example was actually with incoherent illumination. If you had coherent illumination, you can see a little bit of the speckle returning in that image there. So another way in which people have tried to correct for scattering, and another approach that's out there in the literature at the moment that's been picked up on, people are starting to do some interesting things with, is using an ultrasound mediated optical wavefront shaping. So a lot of the problems when it comes to try to correct for scattering and aberration in the sample is that you don't have a known point of reference. You don't really have a way of knowing where your photons have come from. You could put fluorescent beads into your sample, but then you disturb the sample you're looking at, and you don't know that you're always going to have a bead in the place that you're interested in. So this is an approach that tries to get around that, and it tries to use ultrasound to help get around that problem of not having a known point of reference in your sample. So the ultrasound can propagate through the tissue without distortion up to depths of about 100 millimeters. Its resolving power is also determined by diffraction, and because it has a different frequency to that of light, the resolving power of ultrasound isn't anywhere near as good, so it varies from about 20 to about 1,000 microns depending on the frequency of the sound wave that you're looking at. And the point in your sample, if you focus your ultrasound into your sample, the point at which the ultrasound overlaps with the light, you get a refractive index grating. So the ultrasound creates a refractive index grating by moving local optical scatterers, and what that does is it frequency shifts the light that comes into contact with that focus. So what you can do is you can try to observe just your frequency tagged photons, and then you know that those frequency tagged photons have originated from the focus, and in that way you create an ultrasound guide star that can be moved to different positions within your sample, and you can use that to try to kind of retrieve the transmission matrix of your sample via optimization, phase conjugation, sort of different approaches there. And this was in some of the papers, you'll find this technique called TRUE, which stands for time reversed ultrasonically encoded guide star, and the wavefront of the frequency shifted light is measured, and then the opposite correction is applied. So here's some results people were able to achieve here. So that the figure at the top is the experimental setup, so you can see the light beam coming in, you can see your ultrasound transducer here focusing your sound wave within your sample. These kind of sort of yellowy coloured photons represent the photons that have been frequency shifted by the sound wave, and then you use this digital phase conjugation to measure effectively what the wavefront is coming from that sample, and then you can go back and correct that if you want to. So the different images here, so this is measuring the wavefront and applying the associated correction. So it should be said here that because you're limited by the size at which you can focus your ultrasound, the size at which you can focus the sound, that sometimes can impose a limit on the size of the resolution that you can get back. So if you just measure your wavefront and then correct for it, you end up with an image that's now an optical image, but with a resolution that's equivalent to the size of the focal spot you created with your sound wave. If you run an optimization algorithm or an optimization approach instead, you can actually end up with a smaller focus of your light, and that just reflects that there's a Gaussian profile to your ultrasound wave, and therefore your optimization algorithm will naturally concentrate in the center of that focus. And people here have shown that you can get down to kind of 5 to 10 microns resolution with this kind of approach. And these color maps show the frequency shifted speckle field indicating the phase of the correction that's required. So that's a nice approach that's also coming out in the literature. So the other thing I want to cover briefly before we come to the end of this talk is thinking about other ways in which you can also try to get deeper. And another way of trying to get deeper into your sample is changing the wavefront of the light source that you're imaging with. So here's some images here of characterizing some different tissues. So all of this is very tissue dependent. So this is an example of tissue taken from a rat. So you've got a brain tissue, skull, the bone from a skull and skin, and you can see the attenuation coefficients and how they fall off. So these plots here show that the scattering and the absorption, and they combine to give your attenuation coefficients. And this is those combined. And here you have your breast tissue sample. This is a sample taken in breast tissue. You can see these different windows occur, and these windows we often call the therapeutic windows, and they're where your attenuation coefficient is low or where your penetration depth is high. And we often try to target these windows when we're doing our imaging. Because of the sources that are out there that we can typically buy, we tend to look at this first therapeutic window, and a lot of multiphoton microscopes work here. So you're exciting with, say, 800 nanometers, and that allows you to work within this window. But there's a series of papers that have come out recently trying to push the wavelengths longer and access some of these other windows and longer wavelengths and take advantage of the lower scattering properties. So I'd recommend looking at Chris Zhu's work from Cornell University. He's done a lot of work in this area. So this is a 2-photon fluorescence example here, and he's going down to a depth of 1.6 millimeters and imaging with an excitation wavelength of 1.2 microns. So you can see the image he's taken here, and this is of the brain vasculature in a mouse. He's very interested in the imaging of the brain, and that's where a lot of his samples are based. And here's an example of doing 3-photon fluorescence microscopy, as opposed to 2-photon fluorescence microscopy. So the benefit here of switching to 3-photon is that you can use an even longer wavelength excitation, and therefore you can get even deeper into your sample, and you have access to the same fluorophore. So you're exciting the same fluorophore, but you're using longer wavelength light to allow you to do that. And also, because it's a higher order nonlinear process, you also reduce your background. So Chrissy's got a series of papers on that, which I would recommend looking at. But just some final thoughts. I've realised I've gone on a little bit, so I'll just start to wrap up now. So there's a really nice review paper looking at the depth at which we can image in complex scattering material, and it combines these different techniques. And this really goes back to what I was talking about at the start, where we talk about trying to improve spatial resolution and we're trying to improve image depth. And often we can't have it both in microscopy. Often we have to choose, do we care about resolution or do we care about imaging depth? When you think about where we might go in this area and where the kind of next advantages are in the future, you start to wonder, like, where else on this curve can we get to? So can we, for example, produce optical images tens of centimetres deep into biological samples, and what would be the implication of that? Could we look at nano-sized cellular features 1 millimetre deep into biological tissue, and what would we gain by that? What applications would that open up for us? And how far can we push the gap between the two? How far can we image, say, with cellular resolution, and how deep can we get into biological tissue? So really, these are the different areas, and this is where I'm quite intrigued to see where the field goes in the future. And could we make optical imaging competitive in a kind of clinical and medical setting? And just a bit of a caveat on all of that, and I haven't really talked about speed and photon dose that was on my first slide, but if you're trying to make any kind of practical solution with any of these techniques, you do need a sensible imaging speed, and particularly if you want to image a biological sample, you don't want an excessive photon dose either, so that you can keep your sample alive while you're imaging it. That always has to be a caveat in any of these scenarios. So just to summarise, really, I've highlighted some of the work that's happening in this area. There's many other more fantastic examples of groups that are looking to push these limits. So if you're interested, I suggest searching through the literature. What I find quite interesting is that as yet, none of these approaches are particularly mainstream. And what I kind of mean by that is you don't really see any of these wavefront correction approaches implemented as standard on any commercial microscope. So I think there's clearly more work to be done by us in the field to try to bring some of these techniques to a much more kind of practical solution. And it's just to say that we've got a new grant starting in this area. So it's a collaboration between Nottingham, Southampton and the University of Edinburgh. And we have a website and a Twitter address set up, and that's just getting started. So we're going to be looking to push some of these limits of just how deep we can image. So kind of watch this space for what we're able to achieve. Yeah, thank you for your time, and I'm happy to answer any questions you might have. My advice, probably quite simple, is choose something that you enjoy. It's kind of, it takes up a lot of your time research. It's great fun, but if you're not motivated by it and you don't enjoy it, then that will be really hard. So choose something you enjoy, and try to find people you like working with. I think you spend a lot of time with people. I really like collaborative research. I like working in big groups. I like that dynamic of sharing ideas. That's probably not for everybody. So work out what environment that suits you, and try to find that environment for yourself.
Excellent. Anything to add since your talk, Amanda?
No, I think I responded to that question in my talk, is try, even though you're focusing on a specific thing during your study, to try and keep a bird's eye view on the field so you know where you want to go next. But I think what Amanda just brought up is really important, to try and also maximize enjoyment of the present time, because I think at no other time in your career will you have the opportunity, except maybe during the postdoc, to focus in such a way on a single topic, and just to enjoy it, make the biggest bang you can out of that. Yeah.
Yeah, that's really good advice. I often kind of look back and think, like, I wish I could do that again, and just focus on that one specific project and submerge myself completely in it. And that opportunity gets less and less as you seem to progress through. So yeah, make the most of that, definitely.